B lymphocytes, or B cells, are immune cells responsible for producing antibodies. Each B cell produces a unique antibody, which is very, very important for proper adaptive immune function. A helpful analogy for the relationship between an invading pathogen and an antibody could be a lock and a key. Think of a bacterial pathogen like a lock, and antibodies are like keys. If enough keys of high enough diversity are produced, eventually one of them will be able to open the lock. Likewise, if there is a wide diversity of antibodies in a patient's blood serum, there is a very good chance one of them will be able to bind to the invading pathogen and mark it for death by phagocytic cells. A given person's B cell population can produce up to 48 trillion unique antibodies. Now, obviously, our chromosomes are not nearly large enough to encode individual genes for each of these antibodies. So, how then can we produce such diversity? Before we go any further, it is important to discuss the actual structure of an antibody, or immunoglobulin. Each immunoglobulin consists of two identical heavy chain proteins, shown here in dark blue, and two identical light chain proteins, shown here as cyan. The N-terminal ends of a heavy chain and light chain make up the binding site for attaching to an antigen, and the ends of each individual chain are called variable regions. The variable regions, as the name suggests, is what makes an antibody unique. Let's look at how the variable region is encoded for the heavy chain specifically. This is what the antibody encoding chromosomal region, specifically for the variable region, looks like at the germline level. It is completely inexpressible in this form. Notice there are three types of protein encoding segments, V, D, and J. The post-rearrangement gene will only have one of each segment type. The process by which single V, D, and J regions are spliced together is called somatic recombination, and it is only after this process that expression can occur. For the heavy chain, there are approximately 40 V regions, 23 D regions, and 6 J regions, meaning there are 5,520 different combinations for the heavy chain. Light chains have a similar germline structure, except it is simpler in that there is no D region, which means there are 295 different light chain combinations. Altogether, from VDJ recombination alone, there are 1.6 million different antibodies that can be produced. Recombination is directed by sequences creatively named recombination signal sequences, or RSSs, of which there are two types. The first is comprised of a constant heptamer sequence, a 23 base pair spacer, and a constant nonamer sequence. The second has the same structure, except it has a 12 base pair spacer instead. RSSs are the binding sites for recombination enzymes, and also ensure recombination occurs in the correct order. For the heavy chain, a 23 spacer RSS is always found downstream of a V region and upstream of a J region, and a 12 spacer is always found flanking both sides of a D region. Recombination activating genes produce two proteins, RAG1 and RAG2, that mechanically carry out the recombination process. RAG1 binds to an RSS and then binds to another RSS of a different type. The fact that RAG1 cannot bind to two RSSs of the same type is called the 12-23 rule, and it prevents a J region from joining directly to a V region. At this point, the V and D segments to be joined are held in close proximity by the RAG complex. The DNA molecules are then cut at the heptamer sequences, and the segments are joined. The excess DNA that was cut out is called the signal joint, and the joined sequence is called the coding joint. However, if you take a closer look at the mechanics of how the segments are joined, even more diversity is created. The initial cut occurs between the heptamers and the actual V and D regions, and it results in hairpin ends on these regions. The two bases at the ends of the DNA double strands actually share covalent bonds. The RAG complex then makes a random nick in one strand of each segment, creating palindromic single-stranded nucleotides at the end of each segment. These are called P nucleotides, or palindromic nucleotides, and they create more randomness and therefore more diversity. However, the segments still need to be actually joined. This is catalyzed by an enzyme called terminal deoxynucleotidal transferase, or TDT. TDT adds random nucleotides to the ends of each single strand until the two strands can hybridize. DNA synthesis and ligation then fill in the gaps, thereby fully joining the two segments. These additional random nucleotides are called N-nucleotides because they are not encoded by germline DNA. This whole process of joining segments results in the addition of two types of nucleotides, P and N, to the coding joint, which creates junctional diversity. Junctional diversity is an important source of variability in antibody synthesis, and has the potential to increase overall diversity by a factor of 30 million. In all, taking into account recombination from different V, D, and J regions and junctional diversity, your B-cell population has the potential to produce 48 trillion unique antibodies. This should make you feel pretty safe.
Stay frosty, Snowflake.